Good evening, everybody. Thank you for joining our Eversight webinar on novel advances in the cornea and eye banking field, showcase of our 2022 Eversight Research Grant recipient. My name is Onkar Sawant. I'm VP for Research and Development at Eversight, and I'm joined by my colleague, Becky Harris. Becky is our Philanthropy Director for Foundations and Corporations. All right, let's get started. We like to start all of our webinars with our mission. Our mission is to restore sight and prevent blindness through healing power of donation, transplantation, and research. In 2022, for our Eversight Eye and Vision Grant, we had 4 recipients: Dr. Elizabeth Berger from Wayne State University, Dr. Nambi Nalasamy and Dr. Ivanova from University of Michigan, Dr. Sarah Sunshine from University of Maryland, and Dr. Simon Kaja from Loyola University in Chicago. Unfortunately, Dr. Simon Kaja is not going to be here to present his research findings, but we do have other 3 investigators with us. And with that, I would invite Dr. Elizabeth Berger to present her finding. And I'm gonna stop sharing and give control to Dr. Berger to share her screen.
Sure, let's see here.
While Dr. Berger is sharing her screen, I will go ahead and introduce her. Dr. Berger is Associate Professor in the Department of Ophthalmology, Visual and Anatomical Sciences at the Wayne State University. She has been faculty member at Wayne State University since 2010 after receiving her PhD there. Her laboratory predominantly focuses on corneal diseases pathogenesis and interplay between immune and neuroendocrine system. And she's going to tell us about a novel combination treatment to address diabetic-induced impairment of corneal epithelial cell barrier function and integrity. So with that, Dr. Berger, is all yours.
Thank you so much for the introduction, for the opportunity to share some of our recent findings. Thanks to Eversight for funding this work. I'm just going to apologize ahead of time. I have a pretty big dog who is sitting very close to me, so he's very talkative, and so I apologize for that. He's also epileptic, so we got to keep him close by, just keep an eye on him. So if he chimes in, that's what's going on there. But so because we don't have a lot of time, I'm just going to get right in. I won't go over all of the stats that I have here, but suffice it to say that the great epidemic of diabetes is upon us. These numbers are staggering, yet expected to continue to rise in the coming years. And as you know, diabetes can lead to a whole range of health complications, including cardiovascular disease, kidney disease, and vision loss, among others. And although hyperglycemia-induced changes to the cornea can manifest in a number of ways, including alterations to the tear film, corneal edema, epithelial defects, stromal fibrosis, nerve degeneration, many of the complications involve the epithelium. And that is what this project focused on in particular. And so I just listed a few ways in which hyperglycemia can affect the corneal epithelium. Overall, hyperglycemia can have several negative effects on the corneal epithelium, which can then increase the risk of corneal infections and other complications. So what we aimed to do was to combine 2 separate molecules that we've studied in our bacterial keratitis models, thymosin beta-4 and vasoactive intestinal peptide. So I'm just going to provide some information here on thymosin beta-4, or TB4. So it was first discovered in the mid-1980s in an effort to identify small proteins produced by the thymus gland that functioned as lymphocyte growth factors. And as a result, a number of these proteins would go on to become part of the thymosin family, which can be divided into 3 groups: alpha, beta, and gamma. Presently, there are 15 types of beta thymosins with the three main forms found in the human body listed here. And TB4 is the most abundant, accounting for 70 to 80% of beta thymosins. It's produced, TB4 in particular is produced throughout the body, including all cells except for red blood cells, suggesting that it has broader physiological functions than just beyond the immune system as originally identified. So this protein's been shown to enhance the migration of cells to sites of injury, promote angiogenesis, stimulate the production of extracellular matrix components. And because of its regenerative properties, TB4 has been studied for its potential use in treating a variety of conditions, including cardiovascular disease, stroke, skin wounds. Additionally, TB4 has been found to have anti-inflammatory effects. So it's been used or investigated as a treatment for conditions characterized by chronic inflammation, such as rheumatoid arthritis. And so TB4 has been found to have, several effects on the eye, particularly in relation to its potential for promoting tissue repair and regeneration. So I just noted some things in regards to the cornea, the retina. It's also being investigated as a treatment for dry eye, age-related macular degeneration, and diabetic retinopathy. And then the other molecule I mentioned was vasoactive intestinal peptide, or VIP. So it was first discovered in the 1970s by two different groups at the same time. One was at the NIH, and then the other was at McGill University in Canada. And researchers first identified it or discovered it to be expressed in the intestine. And then they found that it was more widely distributed throughout the body and plays a role in a wide range of physiological processes. And today it's recognized as an important neuropeptide with multiple functions in the body, including regulation of the immune system, metabolism, and the cardiovascular and nervous systems. And its discovery has led to numerous studies investigating its potential therapeutic applications. And just like TB4, VIP has also been found to have important effects on the eyes, including regulating intraocular pressure, protecting retinal ganglion cells, and promoting corneal wound healing. And so this is just kind of our basic outline of our experimental design. Like I mentioned, we decided to combine the protective effects of TB4 and VIP as a potential treatment to examine its therapeutic potential in preventing hyperglycemia-induced changes in corneal epithelial barrier function and integrity. And so the data that I'm showing tonight is all in vitro approaches. So we've cultured human corneal epithelial cells under either normal glucose or high glucose, and then treated them with TB4 and or VIP. And we used ESIS, which is electric cell substrate independent impedance sensing, that allows for a non-invasive approach to continuously monitor cellular behavior and integrity. We can also dynamically measure and model different parameter changes in cell migration, barrier function, and wound healing. And then we also carried out Western blot and immunostaining to examine select molecules associated with the tight junction complex. And so what I'm showing here is barrier function assessments. And we measured this by overall impedance shown in this plot tracing here. And this gives us an idea when the monolayer of cells is in place and it's confluent and it's functional. Impedance can then be broken down into resistance and capacitance. So resistance gives us insight into barrier formation and function, and the capacitance gives an idea of overall coverage of the electrode by the cell layer. And we can see this is all as a function of time. And so values were normalized to cell-free electrodes. And then we can see that we have average normalized values that are quantitated in panels D through F. And then areas under the curve are quantitated in panels G through I. And so basically what these results are showing is that all three parameters are significantly impaired in cells exposed to high glucose, which are represented by the dark blue dots, compared to normal glucose shown in green. And that our combination treatment, represented by the fuchsia here, prevented the high glucose induced changes. And these levels are in fact similar to normal glucose, whereas we didn't really see any effect, or at least not the same effect with either monotherapy alone. And so this next slide is showing some mathematical modeling that was applied to derive three different cellular properties. We've got alpha, RB, and CM. So the parameter RB reflects the tightness of the intercellular space. This one in the middle here. And this is highly dependent on cell-cell junctions. And then the two remaining parameters, alpha and CM on either side, are indicative of the current flow below or through the cells respectively. And then panels D through F quantitate total and then endpoint values. And here we see that high glucose, again, significantly reduced the three parameters compared to normal glucose, and that our combination treatment TB4-BIP prevented high glucose-induced decreases in alpha. However, we didn't see any significant differences in RB or membrane capacitance. And here we show cell migration data. Again, indicating that TB4 and VIP prevented high glucose-induced changes in all three parameters. And then now we've got wound analysis. So in this case, we have a high field pulse across the ESIS electrodes that was induced at about 120 hours post-seeding, as indicated by the red lines in each tracing. And we can see that the cells treated with the combination TB4-VIP recovered significantly better despite high glucose exposure, as shown in panels D through F here. And then in panel G, you can see that the velocity of cells migrating to close the wound was significantly increased compared to high glucose as well. So you might notice too in the teal bars that TB4 alone actually fared pretty well as a monotherapy in this wound analysis. But this wasn't too surprising given its strong wound healing potential. But it's also noted too that it didn't do as well as the combination therapy either. And then here we see protein levels for 4 select molecules associated with the tight junction complex. So we've got ZO1, occludin, claudin-1, and ZO2. And while there are no changes in regards to ZO2, we can see that high glucose significantly reduced ZO1, occludin, and claudin-1 compared to normal glucose, and that the combination treatment, again, effectively prevented this decrease. And then this is further supported by immunostaining shown here for ZO1 across the top, and then occludin in the middle panels, and Ki67, a proliferation marker to confirm that the differences observed here weren't due to the cells dying or proliferating at different rates. And these data indicate that the combination treatment maintains a staining pattern similar to normal glucose conditions. So here we've got normal glucose, high glucose, our combination treatment, and then TB4 and VIP. And so we see that, again, our combo looks a lot more similar to normal glucose, whereas TB4 and VIP alone, although we do see some improvement over high glucose, it still exhibits less defined staining for ZO1. And then we actually see a delocalization from the tight junctions with stronger redistribution within the cytoplasm for occludin. And so as a summary of our findings, barrier function, cell migration, and wound healing of the corneal epithelial cells were significantly impaired under hypoglycemic conditions, which as we would expect. But interestingly, high glucose-induced effects on corneal epithelial cells were mitigated by our combination TB4-VIP treatment, whereas either monotherapy alone was not as effective. And so overall, We feel that these results are very promising and supporting further development of TB4 VIP as a combination therapy to protect the diabetic cornea. And then just a thank you to recognition to the lab who does, all of the work, and then for Eversight for the funding.
Thank you so much, Dr. Berger. That was really informative. We'll do question and answer towards the end when all presenters are done. So next presenter is Dr. Ivanova. And Becky, I believe you're going to share the slides. Dr. Ivanova is Research Associate Professor in the Department of Neurobiology at School of Medicine, University of Michigan. Dr. Ivanova's background is in the structural biology, biophysics of protein aggregation and amyloid fibril formation. She joined the University of Michigan Department of Neurology as an assistant research professor in 2013. Dr. Ivanova's lab applies biochemical and biophysical methods to study protein misfolding and aggregation. Today, Dr. Ivanova, who is a collaborator with Dr. Nambi Nalasamy, is going to share how protein aggregation can be affected in endothelial dystrophies and potential for new treatment development. Take it away, Dr. Ivanova, all yours.
Thank you so much for the great introduction. Can you hear me well?
Yes,
we can hear you.
Yeah, thank you. Thank you very much for giving me the opportunity to talk tonight. This is a collaborative project with Dr. Nambi Nalasamy. And here on the two sides, you can see the two buildings where the research is going on. And I want to take my time at the beginning to thank to the undergraduate students, Dominic Ballis and Navralon, who helped with the experiments, and the Eversight Award for providing funds for this research project. Next slide, please. Next slide.
Yep, sorry, just trying to figure out how to, there we go. All right.
So 7 types of corneal dystrophies are caused by the mutations in the transforming growth factor beta-induced protein IgH3. It is also the abbreviated version, which I'll be using through the talk, is TGFBI. This protein is also known as BH3. These dystrophies are autosomal dominant and affect 9 to 15 people in a population of 10,000. Wild-type TGFBI have been implicated in Fuchs dystrophy, and mutants of these proteins are also found to cause keratoconus. The vision decrease, which is associated with the corneal dystrophy, is largely due to the accumulation of misfolded TGFBI in the cornea, in different layers of the cornea, and also there's a lot of scarring due to the recurrent corneal ulcerations or erosions. The current treatment for these conditions is surgical, and they can be roughly grouped in 2 types. The first one is for phototherapeutic keratectomy, PTK, and the corneal transplants. However, with both approaches, both surgical approaches, there is graft rejection, hemorrhages, susceptibility to viral infections in the cases of the corneal transplants, and also double visions and irritability in the case of the PTK. Also, the recovery time from these procedures can last up to a year. And the recurrence and the vision decrease also occurs in the case of the corneal transplants after 4 to 5 years, again, due to the aggregation and misfolding of TGFBI. And currently, there are no medicinal treatments which are available. Can you go to the next slide, please? So our hypothesis is the following. Because of the aberrant aggregation of THGBI correlates with corneal ossification, we believe that stabilizing the native fold of this protein or inhibiting the aggregation of the protein can alleviate the progression of the corneal dystrophies. Can you go to the next slide? So just to give you a little bit of view at the molecular level, how this protein looks like, this is a 683 residue protein and has 5 domains, one amylin or AMI domain at the amino terminus, and this is followed by 4 facilin or FAS domains spanning from mid protein to the C terminus. In blue are shown the locations of the residues which are most commonly found to be mutated in the dystrophies. At position R124 is the mutation typically to cystine, which is very dominant in the Asian population, and the European population. The most common mutations are in the residues of 555, and this is to histidine. Can you click the next one? So what we believe in the case when this protein starts to aggregate, the mutations are causing a local destabilization of the structure of this protein. And as you can see in red, highlighted the regions which are prone to aggregation. These regions are typically hydrophobic, and they are very sticky, and they have very affinity to interact with one another. So in red is they're colored in red. So if they see another exposed region, they tend to interact with each other. And these are intramolecular interactions. This is between the molecules. So in the case when that regions are exposed, they start to recruit like multiple molecules into the growing aggregate, and this is what we believe is causing the pathogenic aggregation of this protein. Can you click the next one? So the routes to the therapy from that molecular view are 2. The first one is find compounds which can stabilize the fold. We will refer to this as a chemical chaperones. And the second one is find compounds which interact directly or transiently with these aggregation-prone regions, preventing them from recruiting other molecules into the growing aggregate. Next slide, please. So our approach is very simple, is to do and develop in vitro assay using recombinantly expressed and purified protein and screen it against an FDA-approved drugs. I want to stress again that this proteins is an extracellular protein, which is also found on the cornea, and this makes it very accessible for topically applied drugs and treatments. And in green are shown the areas, the very early stages on which we have done some progress utilizing the research funds from the Eversight. Next slide, please. So here I just want to spend some time talking about the way we generate the protein, since this is the anchor or the pillar of our studies. We use eukaryotic cells, HEC293, and transfect the plasmids containing the gene of either wild type or mutant protein. Next one. Then this is cultured for about a week. Next slide, please. And the protein is secreted in the media, cell media, and is purified on affinity column using the His stack, which is to which the protein is fused. Next one. And at the end, we do a quality check of the protein. So that step, it took us a while to optimize and develop. So when we started this project, we started it from scratch. And we also suffered quite a bit of the fatigue of SARS-CoV-2 quarantine, as the supplies were taking quite a bit of long time to arrive. But needless to say, we're happy to report that we were able to purify, next slide please, this protein to a sufficient purity ranging from about 92 to 97%. And here is shown the SDS page gels of the wild type and the mutant are the 124C. Can you go to the next one, please? Now, next, we worked on developing the assay for the detection of the aggregates. A molecule called theoflavin T is very sensitive to aggregate formation. So this molecule does not fluoresce when there are no aggregates, but in the case of the aggregation, it produces a characteristic fluorescence. And here showing the formation, the aggregation assays of the wild type THGBI and the mutant R120C co-incubated with the theoflavin T compound, and showing that we are seeing a signal which is suggesting that these two proteins are aggregating. Can you click the next slide, please? And when you view these aggregates under the electron microscope, we were also happy to observe that the mutant R124C forms these very large and very densely populated aggregates compared to the wild type THGBI, where it's the reaction completion, the aggregate, the structures which are formed are much smaller and less abundant. So we are very optimistic that we can translate this assay into the high throughput screens. So the next step is to screen the FDA compounds. And here are shown the heat to lead and lead optimization and preclinical and clinical development, in which U of M has a really huge history and will get a lot of support. I will end it here and thank you very much for your attention, and thank you so much for helping with the slide projection.
Thank you, Dr. Ivanova. That was very informative. Thank you for sharing your findings. Next, we have Dr. Sunshine. Dr. Sunshine, if you want to go ahead, start sharing your screen. And also, just quick note to all the participants, if you have any questions, please post them in the chat, and then we will read those out at the end of this presentation. So Dr. Sunshine is a clinician scientist serving as an assistant professor of ophthalmology at the University of Maryland Medical System. Dr. Sunshine completed her medical training at Johns Hopkins School of Medicine, ophthalmology residency at Northwestern University, and cornea fellowship training at the Illinois Eye and Ear Infirmary in Chicago. She specializes in treating corneal and ocular surface diseases, focusing on complication of oncologist therapy in the eye. more specifically on ocular graft versus host disease. So today, Dr. Sunshine is going to talk about the role of granenzyme B in the pathogenesis of ocular graft versus host disease. It's all yours, Dr. Sunshine. Take it away.
Thank you so much. And thank you so much for the opportunity to share my work with you today, and to Eversight for funding this project. So I'm going to talk to you today about the role of granzyme B in the pathogenesis of ocular graft versus host disease. Maybe. There we go. These are my disclosures. Hematopoietic stem cell transplant is the only definitive therapy for many hematologic malignancies, autoimmune and hereditary disorders. Graft versus host disease is the leading cause of morbidity after a bone marrow transplant, and occurs when the donor bone marrow or immune system attack the host organs, including the eyes, the skin, and the gut. Graft versus host disease affects approximately 50 to 100,000 patients in the United States. So ocular graft versus host disease is the major cause of morbidity associated with graft versus host disease. It affects approximately 30 to 60% of patients diagnosed with graft versus host disease, and begins to affect them about 3 to 12 months after their stem cell transplant. The symptoms of ocular GVHD include severe dry eye, which causes breakdown of the epithelial cells of the cornea and can result in infection, pain, poor vision, and cicatricial conjunctivitis, and ultimately corneal perforation. So despite knowing which patients are at risk of developing graft versus host disease, we're very limited in our treatment options, which are currently limited to lubrication with artificial tears, prevention of desiccation with environmental changes and scleral contact lenses, as well as topical or systemic immunosuppression, which is not a targeted approach and has significant side effects. What's clear in all of this is that we need better treatments and a better understanding of the underlying mechanism of the pathogenesis of ocular GVHD in order to identify novel therapies and better treat our patients and identify those patients most at risk. At the University of Maryland, what we're doing is we're developing a translational research program to study ocular GVHD, which allows us to go from the bench to the bedside, identifying new treatment targets in our mouse models, which I'm going to talk to you about today, as well as utilizing our biobank, which takes patient samples and allows us to study that as well. So granzyme B is a serine protease. It's a serine protease, which has a multifunctional pro-inflammatory properties, which means that it's involved in inducing inflammation by stimulating cytokines release, and is also involved in extracellular matrix remodeling, and has also been implicated in multiple autoimmune diseases, such as rheumatoid arthritis and scleroderma. So granzyme B is part of the auto-amplifying pathway that leads to significant damage in these diseases, including scleroderma and lupus. And it's crucial to, it's been shown in one mouse study, a model of scleroderma that mimics many characteristics of chronic GVHD to be involved in the pathogenesis there via interferon gamma and TGF-beta. So in the eye, granzyme B has been shown to be expressed by the conjunctival T cells, which also allows us to localize granzyme B to the ocular surface So we hypothesize that granzyme B plays a critical role in ocular graft versus host disease pathogenesis via apoptosis, fibrosis, and cytokine activation. It supports its multifaceted effect on the development of ocular GVHD. So to study this, we've optimized 2 mouse models of ocular GVHD or GVHD, systemic GVHD, We have both an MHC mismatched model as well as a haploidentical model or an MHC matched model. Both of these models develop significant ocular findings. And what it is we take the donor's bone marrow from the C57 mouse and then inject it into a lethally radiated host mouse. And that's either a BALB-C mouse, which is a MHC mismatched model, or a B6D2 mouse for the MHC matched model. We then follow these mice for weekly and score them with both systemic and ocular scoring. And we follow them for about 40 to 60 days. So the first thing that we did was we were able to create a new and simplified grading system for ocular graft versus host disease in mice. So what you can see, it mimics our clinical grading system, but we looked at corneal staining, eyelid fur changes, and corneal haze. And what we found was that this was clinically reproducible and relevant to for mice with ocular GVHD. So in our first study, what we looked at was we were evaluating the effect of an IL-2 complex, which we thought would increase T regulatory cells, and we were hoping it would have a protective response. But what we found was that that there was significant worsening of ocular GVHD and GVHD in the mice treated with this IL-2 complex. So, and you can see that at the top. So this is the ocular GVHD score on the left, and on the right is the systemic GVHD score. And the black line is the negative control, and the red and the blue line is the positive control. And the red line is the mice treated with the IL-2 complex. And then you can see the photos depicting the differences. But given that this was an unexpected response, we then looked at the mRNA expression of granzyme B in these in the cornea, eyelid, lacrimal gland, and conjunctiva. And what you can see is that granzyme B was granzyme B expression was significantly elevated in the cornea, eyelid, and conjunctiva, which led us to question whether or not that was something that we should pursue. So high levels of IL-2 is known to lead to an increase in transcription of granzyme B and T cells. So that wasn't particularly surprising, but it did lead us to question whether or not granzyme B was was the cause of the changes that we had seen. So we hypothesized that knocking out granzyme B would result in improvement of chronic and ocular GVHD. So we started with the MHC mismatched model because this is a more inflammatory model than the haploidentical model. And we used granzyme B knockouts both for the donors and the host. And what we found is that again, let me just go through these graphs at the top. So what you can see is that the black lines are the negative controls, both with wild type and granzyme B knockout mice. And then the red line is the wild type host and wild type donor cells. And that is the positive control, essentially. And then what you can see is the blue lines is the granzyme B knockout, both in the host or in the donor cells, and you can see that the granzyme B knockout did have a significant improvement in ocular GVHD and systemic GVHD. Another way to look at this is at the bottom of the slide, so you can look at the severity score, which allows you to see just visually even that the positive control group is significantly more severe as compared to the granzyme B knockout groups. So then we then went on to study this in the MHC matched model. And again, it's the same. We used the granzyme B knockout, both in the donor and the host. And we had found very similar responses. So again, you can see the control groups in black the negative control is in the positive control is in red, and you can see that there's really a very minimal change. There's a pretty significant improvement in both ocular GVHD and systemic GVHD in the knockout groups, and again depicting those changes in the bottom graph as well. So in conclusion, our results suggest that granzyme B may be a therapeutic target for treating systemic chronic GVHD and ocular GVHD specifically. It is a novel target for the treatment of ocular GVHD, and further studies looking at this specific tissue and local effects are necessary to better elucidate the mechanism. And as far as future direction, so the goal of our work at Maryland and the system that we're in has been, the goal is to integrate our mouse models into with our clinical practice and our biobank study. So we're going to evaluate the tiers of our patients in our biobank study, patients with ocular GVHD, and look for changes in granzyme B, and then also look for any potential therapeutic, therapeutic way of targeting decreasing granzyme B topically on the ocular surface. So finally, I want to thank Eversight for supporting this work, as well as my mentors, Dr. Kao, Jang, and Adanakovich, as well as the members of my lab and medical students who have worked with me, and then my other funding sources. Thank you.
Wonderful. Thank you so much, Dr. Sunshine, for that wonderful presentation. And Onkar, if you're ready to share your screen. And thank you also to all of our speakers tonight for sharing your findings and for the meaningful work that you're all doing.
Yeah, I'm gonna just encourage everybody to post the questions if they have any. But I do have questions for our panelists here. So if everybody wants to come back on video, that would be great. Dr. Sunshine, I have a question for you. So when you do corneal transplantation for any of your high-risk GVHD patient, post-transplantation, do you have a different strategy for the care that you provide, considering their immunological status?
So most of these patients don't end up with a corneal transplant, actually. So the transplant that I'm talking about is bone marrow transplant. But to answer your question anyway, I would probably, given that they're immunosuppressed and have, they don't necessarily do as well with the corneal transplant, but I would just use probably higher levels of steroids because that's our only option.
Gotcha. Thank you. Dr. Ivanova, I'm curious, the TGFIP, do you know, are there any studies showing there is age-dependent changes in the protein structure over time, whether it's in human model or any animal model? Just curious if that has already been shown.
So the structure which I have shown, it's just from a recombinantly expressed and purified protein, and you pose a very good question, and this is something which is on a pipeline for us. You can use a cryo-electron microscopy to actually look at the high molecular level and high resolution at the aggregates which this protein is forming. So it has been really very successfully, this method has been successfully used for visualizing the structures of fibrils of alpha-synuclein, which is associated with Parkinson's disease, also tau and amyloid beta, which are the both proteins accumulate in Alzheimer's disease. So this is something which we plan on using the protein for.
I do see a question for Dr. Berger. It's from Sashi Maralupa. They're asking, is the proposed treatment option in your study be topical or systemic or oral? The combination therapy.
Right. So this is topical, actually. Yep. So TB4 actually is in phase 3 clinical trials for dry eye and neurotrophic keratopathy. And so there's an ophthalmic formulation that's already available. And then VIP, we just generate our own topical.
We also received a question in the Q&A that says, for the VIP and TB4, does it exist naturally in tears and aqueous humor? If yes, does its concentration change in diabetes/hyperglycemia?
Yeah, that's a good question. It does, and both of them do, and they do both. Levels do change in diabetic patients. VIP does take a little bit of time to reduce. I think it's like at least in mouse models, it was shown to upregulate and then downregulate about 3 weeks into inducing diabetes. And then TB4, although we haven't shown in human patients that it's been reduced, we do see that there's a reduction in our animal model.
Awesome, thank you. I don't see any other questions. So thank you again. Oh, there's one more question. Would HDAC inhibitor be helpful in diabetic corneal disorders?
That's actually a good question. I'm not familiar with their mechanism. I would have to say that as far as, that's a good question, because I think if I'm not mistaken, it's been shown, those inhibitors have been shown to impact cancer cells through apoptotic mechanisms or being, I'm not super familiar with these inhibitors. But I would imagine, it's a good question because we're not really sure. There definitely is some apoptosis going on with epithelial cells. And so from that standpoint, it might help. But and this is, again, I'm not really completely sure. If they inhibit or if they induce apoptosis in cancer cells. And so in that sense, it would really kind of depend. We don't really want to induce more apoptosis of epithelial cells in diabetic corneas because they're already undergoing apoptosis. And so in that sense, I don't really know. I don't know if the HDAC inhibitors would be helpful. But that's a good question. Sorry, I don't have a better answer.
No, that's great. Thank you, Dr. Berger. And I don't see any more questions. So I'm going to share back my screen. We do have an announcement about the 2023 program. So Becky, do you want to go ahead and mention that?
Yes, I'm happy to share the exciting news. So yeah, it's my privilege to announce our 2023 Eversight Eye and Vision Research Grant recipients. So Dr. Ghulam Muhammad from Wayne State University, Dr. Beth Ann Bennett from Case Western Reserve University, Dr. Victor Guaquil from University of Illinois at Chicago, and Dr. Maria do Carmo Pereira da Costa from the University of Michigan. Congratulations to our 2023 recipients. And for anyone who may be curious, our 2024 applications for the 2024 research grant will be available this summer. So keep an eye on eversightvision.org. That's our website for more details on that coming soon.
Perfect. Awesome. And here's our information. If anybody would like to contact us, has any questions related to our grant program, we are happy to answer any question. So thank you again all for joining us today. And please do reach out to us. And thank you again for our speakers. We really appreciate your work, and it's been a pleasure having you tonight.